the science now, and I'll be talking about how we performed characterization of the antibody response to SARS-CoV-2 vaccination in a macaque model. So one of the things that became apparent in February and March is that there was a dire shortage of non-human primate models for preclinical studies. There have been a few reports in newspapers and other publications that highlight that the need for non-human primates for preclinical studies. And several of contract research organizations and national primate centers have been interviewed for this, and they really highlight the need for non-human primate as a model system, not just for COVID-19 research, but for lots of vaccine and therapeutic studies. And the shortage that is happening in the US was causing a delay in a lot of research. So you may be aware that, of the available non-human primates in the US, priority is being given to studies looking at vaccines and therapeutics for SARS-CoV-2. So this means that other research on other topics is getting delayed. And this is really a problem. The National Primate Centers identified this issue a few years ago, that in the US, we just don't have the resources and the sheer number of animals to support the research that's going on. And right now, as of 2019, the US imported 60% of the non-human primates used in biomedical research. And when China banned all wildlife trade at the beginning of February, that included the non-human primates used in research. So that really affected the supply, but particularly in the US. Of course, there's an increased demand for non-human primate models, as there are over 100 different COVID vaccine studies ongoing right now in the US. So there were pre-existing shortages. This only made the situation worse. And there's not enough domestic supply to meet that demand, and there have been price increases as well. So it's really unlikely that US suppliers are going to be able to fill the gap. So it became apparent that routine screening was going to be recommended to preserve naive animals here in the US that would be needed for vaccine and therapeutic studies in the domestic population. So in July, I believe, Joanne Yee from the California National Primate Center published a paper discussing the broad susceptibility of non-human primates to SARS-CoV-2 and how this could be a problem for domestic colonies, and really highlighted the need for screening. So the Association of Primate Veterinarians and the Pathogen Detection Working Group have been coming up with recommendations for how to do this. They gave a great talk last week on an APV webinar about this, and I've listed some links here in case you're interested in getting that information. But what they recommended is that routine serology or antibody testing be performed to monitor colony status, to make sure that you are maintaining your naive animals, and to perform PCR-based testing only for symptomatic animals or to confirm a seropositive animal. So we saw the need to develop a non-human primate specific diagnostic to use during preclinical studies, that in order to keep the supply of domestic animals safe, we don't want to be competing with the human need for diagnostics. There are millions of tests being performed, and there is definitely a competition for those raw materials, particularly in the earlier stages of the pandemic. People weren't able to get the supplies they needed. So having a non-human primate specific diagnostic will be really helpful, because it's not being used as much as the supply for the human diagnostics. So this will help select naive animals for studies, monitor antibody response during the course of the study, and then of course evaluate the ability, if it's a vaccine study, to generate neutralizing antibodies. And that is the point of what everyone's doing, is they want to be able to elicit a protective response to their SARS-CoV-2 vaccine. The need for diagnostic for colony screening is also apparent, because we really do want to keep these colonies negative for current and future studies, and of course to maintain the health and safety of the animals. So Intuitive Bio, we saw that we had an established antibody platform to address those needs using the colony surveillance assays. we have been using these for years to screen the status of specific pathogen-free animals. And we saw this as a great way to create an assay that would run on the same platform that people were using to regularly screen their SPF colonies and to add in SARS-CoV-2. So I'm going to talk a little bit about how the assay works and then how we used it. So, of course, we have to start off with talking about SARS-CoV-2 virus. The diagnostics used and the vaccines that we used all rely on structural proteins of SARS-CoV-2. So if you're not already intimately familiar with the structure of this virus, the key player is really this spike protein. It's on the surface of the virus particle, and this is what interacts with the host cells. And this has really been the key for vaccines and for diagnostics. Now, many diagnostics also utilize the nucleocapsid protein. And this is on the inside of the virus. It's used to package up the viral RNA genome and protect it from the host response. So similarly, we also focused on those structural proteins of SARS-CoV-2. So as I mentioned, nucleocapsid, it wraps up the viral RNA. It is a fairly simple protein. It's small, it's about 55 kilodaltons, and it functions as a monomer to wrap up RNA. So that is something that we utilize in the diagnostic. The spike protein is more complicated and really a lot more interesting. It is the protein that interacts with the host receptor, the ACE2 receptor. And so here I'm showing some of the features of the spike protein. In general, it's broken into two subunits, the S1 subunit and the S2 subunit. And there's a cleavage site here in the middle that is important for viral entry. And so how this linear sequence translates into a three-dimensional structure is that the S1 subunit forms the portion that interacts with the host receptor and is attached to the stalk. And it takes three full spike proteins to form a functional structure. So it functions as a trimer. The S2 subunit, or the stalk portion, has many hydrophobic regions, which is shown here by HR. And that is what goes into the viral membrane and tethers it to the outside of the virus. So the S1 subunit is the portion of the spike protein that is unique to SARS-CoV-2 and what distinguishes it from other coronaviruses. So this section is unique. This part is conserved. The stalk is really not seen by the host immune response because it is buried within the membrane. And it's also highly conserved because it does mostly function as a structural protein, the structural portion here. Whereas the receptor binding domain is variable between the SARS-CoV and SARS-CoV-2. And that's how you can distinguish between the two viruses. So this is important for us when we were looking at features for immunogens for vaccination. So we focused, as many others have, on the S1 portion that's unique to SARS-CoV-2. And right away, when discussing this with some of the scientists in the lab, we were talking about why wouldn't you want to use the S2 subunit if that's conserved and it might offer more protection amongst the different coronaviruses. So it's not like flu where we have to keep vaccinating and keep vaccinating. And people have thought about this and studied this. And what they found is that this S2 subunit, while it is highly conserved, it's just not immunogenic. So this is really not a portion of the spike protein that the immune system responds to. So this does not make a good vaccine, but the S1 subunit does. So we focused on two portions, what they call the N-terminal domain, and is shown here as NTD, and the receptor binding domain, the RBD. So our hypothesis, of course, was that the receptor binding domain was going to be the most important part for vaccination, because that is where neutralizing antibodies are directed, is to interfere with this interaction here. And if the spike protein can't bind to the receptor, the virus can't get into the cell, infect, and replicate. So here I'm just showing a schematic of the two different vaccine subunits we used. The N-terminal domain, and it's a recombinant protein, and so it is expressed fused to a human IgG Fc. And the FC is just really, it allows proper folding and purification of this protein. Because this does have transmembrane portions to it, the proper folding really, it needs a whole protein to be properly folded. And so by fusing it to this FC, we're giving it this unique portion. It can be made soluble, so it's expressed in cells and then in the soluble portion, and folds properly, and also has proper glycosylation, which is really important in mammalian systems. The same with the receptor binding domain, or RBD. This is also fused to the human IgG FC portion. And so we nicknamed these the NTD FC for the immunization and the CTD. They called it the C-terminal domain of S1. And I apologize, that's a horrible naming because this is the C-terminal domain. But hey, biochemists have their own ideas about naming. Okay. So for the diagnostic, we needed to be able to detect antibodies generated to these two immunogens. And so for the diagnostic, we selected the entire spike S1 subunit. So this covered both the NTD and the RBD, so should be able to detect antibodies to both of these immunogens. The SARS-CoV-2 diagnostic that we have, because we are also trying to distinguish between infected and immunized animals and naive animals, we also include two other antigens in this assay. Nucleic capsid is known to also be immunogenic and is also utilized in human diagnostics. And then we also use the spike S2 subunit because we do think it's important to be able to see if an animal has had a seasonal coronavirus that's not SARS-CoV-2, because that also may interfere with a diagnostic or therapeutic study. So you may want a truly coronavirus naive animal, in which case you'd want it to be negative for all three of those antigens. So just briefly to overview how the colony surveillance assays are made, we take very small volumes of all of those proteins, and we can print them in picoliter droplets into the bottom of a 96 well plate. And so we can individually spot very small amounts of these proteins into unique locations. We spot them in a grid-like pattern in the bottom of a 96 well plate. So when one sample goes into one well, you get 4 replicate results for each one of these 5 or 6 antigens that are present inside the well, including some controls. So generally how it works, the different portions of the antigens are immobilized to the surface. You add in serum from the animal that you're screening. It's diluted serum. It's about a 1 to 100 dilution, and you leave it on for an hour at room temperature. You wash it off, and any unbound antibody goes away. If there are antibodies to those antigens, they'll bind to the surface here. And then we add an anti-simian IgG gold antibody, and this is how it's detected. Again, that's incubated for an hour, washed, and then the detection reagent, the silver quant reagents are added in, and this is what develops a gray signal and how we're able to read the assays. So what that looks like, these are real images from wells of the SARS-CoV-2 assay. A positive sample will have dark signal on the spots for spike S1, spike S2, and nucleic capsid. A negative sample will have nearly no signal. This animal looks like it has a little bit of S2, which is not unsurprising because there are seasonal coronaviruses out there. So this is what real results would look like. We use an instrument or an analyzer called an AQ1000. It takes a picture of the bottom of the well and then basically captures how dark these little circles are and assigns a number to it. The darker the spot, the higher the relative intensity unit. So you're going to see a lot of graphs with relative intensity units on it. And what we're looking for is, the darker the signal, the more antibody is present on the surface. So instead of just laying around being lazy during our time during lockdown for SARS-CoV-2, we got together with 3 other companies and decided to study a recombinant subunit vaccine for SARS-CoV-2 and use the CSA kits to monitor the antibody response over time. And this was a really exciting study. We had 2 commercial partners who approached us asking how they could help and take part in this study to create a diagnostic tool that would be helpful for the rest of the biomedical research groups in the US. And I thought that was a really awesome response to the need out there. They saw that they thought it would be interested to be able to distinguish between infected animals and vaccinated or naive, and develop a tool to screen for naive animals for preclinical trials. Also, to be able to perform antibody titers if you are doing a vaccine study. So, just to refresh everybody, the recombinant proteins we used for immunization were the N-terminal domain NTD FC and what we called the CTD FC that has a receptor binding domain. So we're going to call it NTD FC, CTD FC, and then the entire spike S1 is what's used in the diagnostic. Here's a little bit of information about those immunogens. They are expressed in Chinese hamster ovary cells, so they are glycosylated, and the immunogens were affinity purified. So this is the design of the study. The plan was to hyperimmunize with the SARS-CoV-2 spike. We had a total of 6 animals, 3 groups. All the animals were male. They ranged in age. I think they were between 2 and 7 years old, and they were randomly assigned to groups. Group number 1 received the N-terminal domain FC fusion, and group 2 received the C-terminal domain FC. Group 3 received a mix. And so for each one of these groups, we had a total amount of 250 microgram of recombinant protein plus adjuvant. And this was all made just immediately prior to injection. So you'll notice here that this animal has 3 syringes while the rest have 2. And I want to point that out because the plan had been to immunize all the animals 3 times. We weren't expecting this to give a really robust response. And so we were expecting to need an initial vaccination and 2 boosts. And so we had planned to do the boosts 2 weeks apart. And we did see, and you'll see with the results, that we did not need that 3rd boost for most of the animals. So here's the overall plan. The study was planned to go on for 12 weeks. We would do a blood draw at time 0, collect serum, and then immunize with the first dose. Wait 2 weeks, collect another blood draw to get our 2 weeks post-immunization, and this would be a single vaccination event. And then we'd give them the 2nd boost, collect at 4 weeks. And so this would be, every animal would have had 2 boosts here. And then we'd collect serum every 2 weeks on through 12 weeks. At this 4-week time point, the study coordinator decided that we would pause the vaccination because we were getting a really robust response, and I'll show those results in a second. But also there was some slight injection site reaction. So we decided that we would wait to boost the animals and see if the antibody started to decrease, or if we could just keep it at 2 doses. And so we decided to dose this 3rd animal. That was the only one that needed another boost at 6 weeks. So this was kind of an on-the-fly decision as we were doing this study. And we ended up being lucky enough to go all the way out to 20 weeks for a final collection. Again, we weren't expecting the response to be so robust. And we were lucky enough that the animals were still available to collect at 20 weeks. So again, these animals were immunized with these 2 different immunogens, and then we're going to be using the diagnostic shown here to look at antibody levels to spike S1. Spike S2 is also on the diagnostic, but the animals are not immunized to it, so they should not have antibodies to it. And what this looks like, this is the quick answer, that we took serum at 2 weeks and compared it to the 0 time point, the pre-bleed. And just you can look, visually seeing here, you can see that at the pre-bleed, this animal had no antibodies to spike S1. And 14 days later, it had a really robust response. And we'll see the numbers for this, but I think it's just visually compelling to see that even at 2 weeks later at 1 dose, we have a very, very strong response. And this is actually maximum signal on this assay. So this was very unexpected and very exciting for us to see. And this is just an example of one animal. This is the animal that got that C-terminal domain, cTDFC. And remember, that contains the receptor binding domain. So we were very excited about this. So then we looked across the time course, and I'll show you the time course in a second. I just wanted to show you the magnitude of the response of the immunized animals. So here on the y-axis, we're looking at relative intensity unit. And again, that corresponds to how dark each one of those spots are. We're looking at the signal for spike S1. So this is looking at the relative amount of antibody to spike S1 in immunized animals. I'm showing here a group of almost 200 negative animals. We're considering these all negative because they're from our serum banks, and they're all pre-2019, so they should not have seen SARS-CoV-2. And then in our positive set in red, these are all the animals that were immunized to spike S1. And this is all 3 groups are shown here with multiple time points. In green, you're seeing that we have 5 human serum samples. This is convalescent serum from mild to moderate disease in humans, just to show you, the spread of antibody response. The cutoff value for our diagnostic assay is shown here. And so all the immunized animals are above that value. And are giving a fairly strong response. Same thing for, in this assay, of course, we're looking at spike nucleocapsid and spike S2. So the immunized animals have no response to nucleocapsid, 0 antibody there. Spike S2, this is absolutely what we would expect to see, that there are animals prior to 2019 that have had seasonal coronaviruses. So you do see high signal in some of those animals. And of course, in the humans that did have SARS-CoV-2, the COVID-19 positive samples, they are positive for all 3 antigens, the spike S2, nucleocapsid, and S1. So when we look at our 6 immunized animals over the time course, this was really the exciting information that we got, was that our 3 groups here, the NTDFC, cTDFC, and then the mix. And remember that these 2 groups are the ones immunized with the receptor binding domain. This is not. We see a weaker response in this group 1, but they have high antibodies throughout the course of this study. And the other 2 have very robust, early, and strong lasting antibody response. So this was really exciting, but we are doing this at a 1 to 100 dilution, and this is almost maximal signal for this assay. So we thought that this could be obscuring some of the differences between the groups. And so we wanted to do antibody titer. And so when we do an antibody titer, I'm sorry, we're going to look at immunized and infected before we look at the titers. Here I'm showing the 12 weeks post-immunization, all 6 animals, and these are all 3 of the antigens. Spike S1 is in blue. So for these animals, you really don't see any signal for nucleocapsid or spike S2. For the humans who are infected, you see high signal in all 3 antigens. So this is a really nice way to distinguish infected from immunized by having this multi-antigen assay. You're immunizing for spike S1, and it has a clear, strong response, and it's very different than those that are infected. The other thing we wanted to point out is that looking at this profile for the hundreds of naive macaques that we screened, there are plenty that have signal to spike S2. There are some that also have signal to nucleocapsid, and I'm showing their respective cutoff values by the dotted lines here. correspond with the color. So one of the important things to note is that there are commercially available ELISAs out there that are using a mix of spike S1 and S2 as the antigen. And you can imagine if that's the case, that all these animals with high S2 signal are going to give false positives. And I think that could be a very frightening thing for a group who's doing colony management to think that they maybe have 20% positive animals. It's not the case. It's signal due to S2. So we really think having a multi-antigen approach is better to be able to see the differences between a seasonal coronavirus infection and a true SARS-CoV-2 infection. Okay, here's those titers I was talking about. When we do an antibody titer, we're using the same CSA SARS-CoV-2 assay, but we're doing a serial dilution. And so we start with 1 to 100, and then we dilute it out. We stopped the dilution at 256,000 because it really is going way out there. And we were surprised that our two groups that received the receptor binding domain immunogen, they all had titers greater than 256,000 by 12 weeks. And of course, the group 1 that received the N-terminal domain, they had lower titers. The two different animals, of course, had a different response, a slightly different response. But I think that this was important to see that our hypothesis that the immunizing with the receptor binding domain would make a better vaccine. And that is what we're seeing at 12 weeks post-immunization for sure. And I'm showing the individual animals here just so that you can see, the individual response and it's not being obscured by error bars. So we did the antibody titer using that same type of serial dilution throughout the time course. So I just wanted to show you from 2 weeks at 1 dose, 4 weeks, 1 dose, and then 10, 12, and 20 weeks. So you can see that group 1 have a very low titer after that first dose. After 4 weeks, they have a higher titer. They still have just 1 dose, but really after they get that boost of the second and third doses, they have a much higher antibody titer. And I've put the star here by this animal, animal 1. This is the animal that got that third boost. And you can see the difference that third boost gives. That animal got boosted at 6 weeks. Animal 2 did not. And so this one really does have a nice high antibody titer. But what's surprising is that by 20 weeks, the titer is down again. It's really not that great. So this N-terminal domain FC, this immunogen did not work as well for vaccinating. But both groups 2 and 3 that received the cTDFC that contains the receptor binding domain, those all had fairly high titers at 2 weeks and 1 dose. We were really surprised by this, how large these titers were. And then they maxed out at 12 weeks. And at 20 weeks, I'm highlighting this because that's where we're actually able to see some differences. Unfortunately, this animal, we were unable to sample at 20 weeks. So I don't have the results from this animal. It had been reassigned to a different study. For animal 101, it has a nice high titer, same with animal 202. This animal is starting to lose some of the titer, but we were really encouraged by these results because that having a lasting antibody response after 4 months is really critical. So this was very exciting for us and for the group that is making these recombinant protein vaccines. So the last thing that we really needed to show is that now that we've immunized them with this receptor binding domain, and that's what we thought would make neutralizing protective antibodies, can we actually show that this immunization prevents binding to the ACE2 receptor? So we do not have a BSL level 3 lab to do actual virus challenge studies. And that was beyond the very generous donation of time and labor that the groups collaborating on this study were able to offer. So instead, we were able to use a neutralizing antibody assay that many are using as a surrogate to actual live virus studies. And so what we do is we use the same platform, the colony surveillance assay platform, but instead of printing viral antigens, we deposit ACE2 receptor on the surface of those 96-well plates. We then mix in serum from the animals with recombinant spike protein. And if those animals have successfully generated an antibody response to the receptor binding domain, it should compete with ACE2 for binding to that. So if those antibodies from the immunized serum bind to the receptor binding domain in the exact region that the ACE2 receptor also binds, it should compete for binding to the receptor binding domain. So we would see here, this is a competition assay. If you have neutralizing antibodies, you'll have a lower signal in this assay. So if you do not have neutralizing antibodies, all that receptor binding domain is going to bind to the ACE2, and you'll have nice dark spots. If you have neutralizing antibodies, they're going to outcompete away that receptor binding domain, and you will have lower signal. So that's what we're looking for, is a decrease in signal. So we hypothesized that our groups 2 and 3 from our vaccine study would have neutralizing antibodies because they were immunized with the receptor binding domain. And here's what that looks like visually. Here's animal 101, and this was the animal that was immunized with the receptor binding domain. So it's pre-immune or 0 week. We have 2 different concentrations of this ACE2, and you can see that there's signal there. After 8 weeks immunization, it's gotten 2 doses of this vaccine, you can see that that signal is gone. It has completely inhibited at this concentration, and I think this is about 90% inhibition here. So this is the animal that had neutralizing antibodies because it was vaccinated with CTDFC. In comparison, the animal 01 at 8 weeks who was vaccinated with that N-terminal domain, look at that, no inhibition of binding to ACE2. So this is no neutralizing antibodies. And what that looks like numerically when we calculate maximal signal in the presence in the pre-immune serum, we can see that at 8 weeks, that animal 101 has over 90% inhibition of binding to the ACE2 receptor. And this was similar to a mouse monoclonal antibody that is commercially available and is a known neutralizing antibody for SARS-CoV-2. So this was really exciting for us, and we're definitely doing more work at characterizing exactly this response, and of course the time points at 12 and 20 weeks post-immunization. But this definitely shows that immunization with the CTDFC generates neutralizing antibodies to SARS-CoV-2. So that was a very exciting study to be a part of. We saw that the CSA SARS-CoV-2 assays were able to be used to evaluate the generation of the antibody response to immunization with recombinant protein vaccines. We saw antibody at 2 weeks post-immunization, and I wish we could have taken an earlier time point. You know, hindsight is 2020, but it was a very robust response with 1 dose at 2 weeks. 2 doses elicited an even stronger response that lasts beyond 20 weeks post-immunization. We were also able to use the CSA platform to develop a neutralizing antibody test. And so this is how we're able to show that immunization with the C-terminal domain or the receptor binding domain can inhibit binding to ACE2. And so we are working with a couple other groups that are performing vaccine studies with these same immunogens to work on dosing, timing, how far apart should the doses be, and with the ultimate goal of looking at maternal transfer of antibodies in pregnant macaques to see if immunizing pregnant mothers will then also protect the infants. And so that work is ongoing now. I believe the grant application for the maternal transfer is being reviewed in 2 days. So this is all very timely and up-to-date work that is going on. And I did want to mention that the SARS-CoV-2 test that Intuitive Bio offers, we tested lots of different species of non-human primates because, again, in the beginning of the pandemic time, when we're all at home trying to stay safe, um, we didn't know which animals were going to be susceptible or not. And we tested all the animals we could to see if our test would work, if anybody had antibodies to them. And then over the months, we saw that really macaques are definitely a good model for experimental infection, but really the African green, um, those are even better. They exhibit a disease severity that's similar to humans, whereas macaques get a much more mild disease. And so I would think that people who are in charge of colonies of African green would have more urgency to do more routine testing because they really could have more severe disease. There also is one case of naturally acquired infection in those African green monkeys. But I would say also zoo populations might be interested where you have that interface of human and animal interaction, and people are not very well behaved at zoos, and there can be a lot of transfer of material, food, other waste that can go back and forth. So we are definitely interested in testing all those species. So far, we have not seen any positives. With that, I wanted to point out the collaborators and kind of highlight their roles. Intuitive Biosciences developed all the assays used in this work. Lytix Solutions is also located here in Madison, Wisconsin. These two individuals developed the recombinant proteins, the spike proteins that were used for the vaccine work. And they did an amazing job of cloning these proteins in record time, producing large amounts in order to do a vaccine study and supporting us throughout the whole process. And of course, Covance and Novartis. Novartis team supported the study design and helped supply the animals for this study, while Covance administered the study and actually did the immunization and the preparation of the vaccine work. So this was really a lovely collaboration to be a part of, and we were all calling in weekly from our various homes to discuss the progress. And with that, I just want to open up if anybody has any questions. I can unmute you, or you can unmute yourself, and we can chat about anything that I presented, or if you have a question you want to ask, that is fine.